Okay, we're going to make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are going to be looking at vaccination. So in our last lesson, we looked at photosynthesis, which was a year 13 topic. And as I mentioned, we're flipping between year 13 and year 12 topics every alternate weeks. And in today's lesson, we have come back to the topic that we were looking at previously. So we're on immunity, and we looked at agglutination and antibodies in our previous immunity lesson. Today, we're going to be looking at vaccination. And once we've done vaccination, we've basically done the majority of immunity, and we can move on to looking at monoclonal antibodies, which is again a really common topic that is assessed at A level. Okay, so we're going to go through sort of the content. There are some key points I'm going to mention. Many people struggle with this aspect of immunity. Vaccines is pretty much the same as GCSE. However, there's a key aspect of passive immunity and active immunity that many people tend to get confused with, and also herd immunity. These three things are focused on a lot at A level. That's what we're going to focus on in today's lesson. And then once we've had a look at that, we're going to move on to some exam questions. So looking at the specification, again, we've covered most of the specification. We've looked at the immune response. We've looked at all the first part of it, and scrolling down, we've looked at all of the antigen stuff. We've looked at antigen presenting cells. We've looked at the immune response, the cell mediated response, the humoral response. We've looked at antibodies. We've looked at agglutination. We've looked at plasma cells and memory cells. The only thing that we haven't looked at is the small part at the end of the spec. We've not looked at the use of vaccines to provide protections for individuals and populations against disease, and the concept of herd immunity, the differences between active and passive immunity. That's what we're going to be looking at in today's lesson. Okay, at any point, if you have any questions, you can let me know in the Q&A. I'll be more than happy to answer them for you. So we're going to start off this lesson, and we're going to look at vaccines to start off with. And if we think back to GCSE, the definition for vaccines is the exact same for A levels. So vaccines, they contain small amounts of weakened or dead pathogen or antigens that are introduced in the mouth or by injection. That's the definition for what a vaccine is. So remember, the purpose of a vaccine is a vaccine has these pathogens, these weakened or dead forms of the pathogens. That depends on the vaccine that you are receiving. It could also contain proteins of a pathogen or specific antigens from the pathogen, and that is basically introduced within your body. And because it's in a weakened or dead form, or it's only containing a part of that pathogen, that means that it can't sort of really sort of harm your body in sort of a detrimental way. It can only sort of maybe give you some flu-like symptoms or some minor symptoms that you would be able to cope with. And in return for that, it will produce your antibodies, it will produce the memory cells within your body. So then if that pathogen was to attack you again in the future, your body would be ready to fight it off. So it basically gives you immunity without you actually getting the disease itself. Okay, that's the purpose of a vaccine. So what happens is that when the lymphocytes, so remember you've got your B cells in your body, you've got your T cells, when they are exposed to these antigens, they are activated. The B cells within your body, they are activated. Remember, the B cells, they are part of your humoral response. The T cells are part of your cell-mediated response. So these lymphocytes, the B cells, they respond to antigens that are floating around in your bloodstream. So the B cells, they will look at these antigens and they will say, okay, this is a foreign antigen. This doesn't belong in the body. And because these antigens are the weakened form of the pathogen, they're not really going to affect your body as much. So what these B cells do is they will go and bind onto these antigens, and they will undergo clonal expansion once they've been activated by a T cell. Okay, so remember, T cells are the ones which are part of the cell-mediated response. They will respond to the antigen present on macrophages, on phagocytes, and they will activate the B cells. The B cells will have the antigen present on their surface, and they will be activated by the T cells. So once the B cell has been activated, it will go through clonal expansion. Okay, so B cells then undergo mitosis and they produce a large number of these antibody cells. Okay, they produce a large number of these cells, and then these cells can either differentiate into plasma cells or they can differentiate into memory B cells. There are two types. Again, we've already looked at them. Plasma cells, they are there to produce antibodies, and the antibodies are short term. Okay, so plasma cells are only effective in the short term, whereas your memory B cells, they are there for decades. They live for decades within your body. Okay, they are there in case this infection ever infects you or enters your body again in the future. Okay, so that's what vaccines are overall there to do. In the end, your goal is to produce plasma cells within your body and memory B cells. And the reason why it says all memory B cells, it depends on the vaccine that you are given. Okay, so for example, there has been a question on this in the past, and I'm using the same context from that question. Let's say that you've been bitten by a snake. Okay, let's say a snake has bitten you and has passed on the venom into your body. Now, if you get vaccinated against the venom, okay, and you're given the vaccine that will produce memory B cells, the memory B cells will take time to get produced and they are long lasting within your body. Whereas plasma cells, they are more quickly produced and they are short term basically. So we would need a vaccine that would trigger our plasma cells to be produced fast. We don't care about memory B cells, we just need that venom to get out of our body. So the plasma cells, they will produce the antibodies and they will counteract the venom that the snake has sort of bitten us with. Okay, and the better context will be provided in the exam question. I'm not sure if we've got it today, but if we do have it today, I think we do have it today. So we'll have a look at it towards the end of the lesson about the concept of the venomous snake. Okay, so that's basically your aim of vaccination. Vaccination is really important to counteract any diseases because vaccines will contain the weakened or dead form of a pathogen or antigen and it is injected within you so your body can trigger the immune response. Okay, so moving on. The next part of the spec talked about passive immunity and active immunity. This is everything that you need to know about vaccines. You also need to know about your primary response and your secondary response. But again, that is pretty much the same as GCSE. And what that basically is saying that when you have a vaccination, let's say you get infected with the vaccine gives you the pathogen, your antibodies in your blood slowly rise, okay, to get rid of the pathogen and to produce memory cells, and then it goes back to its original level. And then, if you get infected with the same pathogen again in the future, or the same infection again in the future, your body will produce much more antibodies at a much faster rate, okay, to prevent you from getting ill and getting rid of the pathogen from your body as quick as possible. So this is the graph that you would normally see. This is your primary response, and this is your secondary response. As you can see, in your secondary response, you see a much faster, much steeper curve, much faster production, much faster rate of production of your antibodies. You also see many more antibodies being produced and at a much faster rate. Okay, to get rid of that pathogen from your body as soon as as soon as possible, because your body already recognizes that pathogen. Okay, because you've been vaccinated against it. So moving on, looking at passive immunity. Now you need to be able to compare between passive immunity and active immunity. Now, passive immunity is immunity where you don't create the antibodies yourself. Okay, so you can have antibodies introduced to your body, you can have antibodies injected within your body, or your body can do the work and produce the antibodies itself. Okay, so let's say you got COVID. It is unlikely that you're going to get COVID again very fast. Okay, and the reason behind that is, let's say you've had COVID, your body has produced antibodies against the COVID virus. The antibodies are present within your body. So if COVID infects you again, then the antibodies will be really fast at counteracting the COVID virus which comes into your body. Okay. Now, passive immunity, you're not doing the process. You're not actually creating, your body isn't creating the antibodies itself. Antibodies are introduced into your body. Okay, they are introduced into your body, and an example of how they can be introduced could be through an injection. An injection of antibodies for the COVID virus can be injected into your body, and then the antibodies come into your body and they will just get rid of any sort of COVID particles that are lurking around within your body. Now, the advantage of this passive immunity that you have is that these antibodies will work very fast. Okay, because literally the antibodies have been made, they've been created already, they're ready to go, and they've just been injected into your body. They will be very fast in acting and they can get rid of the virus particles that are present within your body. However, when you're injecting antibodies in within your body, the plasma and the memory cells, they are not created. Okay, they are not created as your body didn't naturally make the antibodies. The pathogen hasn't physically entered your body. So the body doesn't know what antibodies to create and for what antigen. Okay, so if you just inject antibodies into your body, you might be able to get rid of the virus that is lurking around within your body. However, that virus can again attack you in the future. Okay, or if not all of it has been removed, it can continue multiplying within your body. This is only done in cases where the disease is a bit sort of is contained with only one part of your body. It's not really spread through the body, or it's at its early stages, or it's sort of it can cause death. Basically, it's only done with diseases like that. So if we looked at the venomous snake example, let's say if a person has been bitten by a venomous snake, passive immunity will be used, a form of passive immunity, such as injecting in antibodies into the blood to get rid of the venom from the snake, would be used rather than active immunity, because passive immunity is much faster because you're injecting in the antibodies straight into your blood. Okay, and as with passive immunity, no long-term immunity is formed because you haven't physically produced the antibodies yourself. Your body hasn't worked to recognize the antigen, and therefore the antibodies haven't been produced, which means that in the future, that same thing can infect you again. So that's what you have in terms of passive immunity. You don't create the antibodies yourself. Okay, they are just naturally passed within your body. That's a way that you can use to remember passive immunity. The antibodies are just passed into your body. The other type that you have is active immunity. And active immunity, you are actively creating the antibodies yourself. That's what active immunity is. And that's a way that you can remember active immunity as well. So passive immunity, antibodies are being passed into your body, whereas in active immunity, you are actively creating the antibodies within your body. Okay. Now, if you're creating antibodies within your body, that means you will have immunity because your body is creating the antibodies itself. Okay, because you've either been exposed to the pathogen or its antigen. Okay, and there are two types of active immunity that you can have. And the two types, you can have natural active immunity or you can have artificial active immunity. Now, artificial is man-made. So vaccines, they are man-made. Vaccines are an example of artificial active immunity because you're introducing a weakened version of a pathogen or its antigen within the body. So that's an example of artificial active immunity. You could have natural active immunity. Now, natural is you do it yourself. So you could have contracted the pathogen yourself just naturally, and your body triggers an immune response. You might feel ill for a period of time, but your body is building those pathogens. Your body is making those antigens, the antibodies against the antigens from the pathogen. So that's natural active immunity. Your body is actively working just naturally. And then by doing that, your body creates its own antibodies and its own memory cells. Okay, so in the future, if any pathogen like that enters again, you would be immune to that pathogen. So a common example includes the flu. If you have the flu, it is unlikely that you're going to get the flu again and again within the span of maybe weeks, because once you've had the flu, your your white blood cells would be producing the antibodies against the flu virus, and your body would be immune to the flu virus for a period of time. Okay, let's say after a year, then if you contract the flu, you might feel ill once again, because it's been a while since you've contracted the flu. That's why for older adults, you've got flu jabs which are available, because obviously when you are older, your immune system is a bit weaker and your immune system can be more susceptible to opportunistic infections. So that's why flu jabs are available for older adults, to make sure that they don't extract the flu, and they are available maybe every 6 months, okay, because older adults tend to have weaker immune systems. So that's your passive immunity and your active immunity. You need to know how to compare between the two. And I've picked out a 5-mark exam question as well, or a 4-marker. I think it's a 5-mark exam question. So we can compare passive immunity and active immunity, and you know exactly the points that you need if you're asked this question in an exam, because that's a common exam question that is asked. So passive immunity, the antibodies are just passed into your body isn't actively making the antibodies, and the antibodies are there to fight off any pathogens present within your body. No long-term immunity is formed. In terms of active immunity, your body is actively making the antibodies. And because your body is actively making the antibodies, that means you will have long-term immunity. Now, there are two types of active immunity that you can have. You can have artificial active immunity, or you can have natural active immunity. Artificial just means that Artificial just means that it's man-made. So vaccines, for example, is artificial, and you're introducing the pathogen or a dead or weakened form of an antigen into the body. And then you've got your natural active, where your body has naturally contracted the pathogen and is creating antibodies against it. Okay, so that's natural. That's your passive and active immunity that you need to be aware of. Now, what we're going to do, you don't need to explicitly know about the next stage. However, I think it's good to know in terms of the types of vaccines that you can have. So the types of vaccines, these are the 3 types of vaccines that you can have. You can have an attenuated vaccine, you can have a dead microorganisms vaccine, or you can have isolated antigens vaccine. Okay, so those are the 3 types of vaccines that you could be given. Now, if we think about an attenuated vaccine, this is most vaccines. This is where the pathogen is added, however, it's been weakened. Now, because it's been weakened, that means your body won't experience as extreme side effects. Okay, but it won't be suitable for those who have weakened immune systems. So it won't be suitable for those who are older. It won't be suitable for people who have immune diseases, that sort of stuff. However, it will be very useful in terms of providing long-term memory cells within your body. You have the dead microorganisms where the pathogens are killed, but the antigens are present, so your body can produce its own antibodies and its own plasma memory cells as well. However, because the microorganism is dead, this doesn't provide long-term immunity. And then you've got isolated antigens, where the proteins are isolated from the surface of the pathogen to prompt a specific response. Again, this doesn't provide long-term immunity for the isolated antigens. They've literally just taken proteins from the pathogen's surface, if there are proteins, and they've injected it within you, because so you have some form of an immunity, some sort of immunity within your body against that pathogen, but no long-term immunity will be formed. And the final concept that you need to be aware of is herd immunity. Again, herd immunity is a common concept and it can be asked on exam. As we saw on the spec, it is mentioned explicitly on the spec as well. So in terms of herd immunity, I think you might have looked at this at GCSE as well, but if enough of the population are vaccinated, the pathogen cannot spread easily amongst the population. And you've probably looked at this diagram. Let's say you've got these people over here standing around each other. Okay, there's these people, but let's say that the shaded ones are vaccinated. Okay, the shaded ones have been vaccinated, and the ones which haven't been shaded aren't vaccinated. As you can see, they are not in contact with each other as much, which means that they are unlikely to get the pathogen because they are surrounded by people who don't have the pathogen because they are vaccinated. And the purpose of herd immunity is to provide protection for those who are not vaccinated. So some people will have objections against vaccination. Some people won't want to get vaccinated against certain diseases. And for those people, if the majority of the population is vaccinated, then they will also be protected, because that means that the pathogen can't sort of move easily amongst the population. Okay, and other examples that you can have is that those who are too ill to have a vaccine might not have a vaccine, and children who are young might not also be suitable for having a vaccine. Okay, so that's the concept of herd immunity. Again, something that you may have covered at GCSE. Okay, so that's the concept of herd immunity. And in terms of content, that is everything that we're going to be looking at in today's lesson. And from the specification, we've looked at everything in terms of immunity. So we've looked at the use of vaccines, we've looked at the concept of herd immunity, and we've looked at the difference between active and passive immunity.